And the funding is coming from the National Science Foundation largely, through the Center for Hierarchical Manufacturing at UMass and the Materials Research Center on Polymers at UMass, in addition to the Center for Integrated Quantum Materials at Harvard and MIT, as well as community-based learning at Amherst. I would like to introduce Sherman to take it from here.
All right, great. Thank you, everybody. So as you've all seen, we're going to be talking about fighting disease with code. So my background, I'm a physical chemist by training, meaning I study chemistry and physics and the intersection between chemistry and physics. And then in grad school, my professor, who was a physical chemistry professor, ended up moving into the computer science department for his tenure. And that was actually at MIT, and there's a bunch of cool robotic stuff going on around there. But really, what we're doing is this intersection of the world of computer science and the world of chemistry, and trying to use computers to simulate what's going on at the molecular level. So does anybody know what this is? Somebody in the back, I heard? Ebola. So very popular in the news today, right? So people see pictures a lot like this, and you think that this is, you don't know what it is. You see a stuffed animal that might be a stuffed Ebola, and you try to understand, how are you going to fight this disease, for example. So if you start to take a closer look, you start to see that there's actually real structure that's going on in this thing. It's not just this, fuzzy little animal, but it's this thing that's got these little, bumps and grooves and all this other stuff. And if you start to look even closer, you start to see that at each one of these little things is actually a real structure. It's actually a protein. And so proteins are everywhere in the body, obviously. And there's protein networks that are basically all these proteins communicating with each other. And that's how your body functions. That's how cells function. You have all of these very complex networks of proteins and small molecules and vitamins and sugars, and all of these things are communicating with each other based on how they're interacting. And so right here you see actually a picture of a slice of this Ebola virus, and then a part of it, the protein structure, and this is actually some antibodies that are bound to this. So they found a person who was immune to Ebola, and they found that this person's body had actually generated these antibodies that detect the Ebola and clear it out of the system. And so this person doesn't have any response to Ebola. He can't get hurt by Ebola. And then we could start to look at these types of things, and we could start to learn how this antibody is binding to the protein in the first place. What are the actual underlying properties associated with that interaction, and how can we use those interactions to start to develop drugs that can actually be used to protect other people who are not immune to Ebola. So taking a closer look here, and sometimes some of us have seen these types of pictures. They look like these kind of pretty fuzzy representations of these blobby things. And what I care about is stuff that's happening kind of at this level. And so we're simulating proteins, and we're simulating atoms that are interacting, or molecules that are interacting with these proteins. So we're simulating atoms and molecules. And so I think we all kind of have a sense of where atoms lie in the grand scheme of things. Atoms are things like hydrogen and carbon and nitrogen. As you start to look at combinations of molecules or atoms, that they start to come together, like some of the things we have up here, you start to get proteins and DNA and RNA and vitamins and drugs. All these are molecules, and as you start to put molecules together, you start to get cells, and as you start to put multiple cells together in some kind of organized system, you start to get organs, and at the end you actually get organisms. And below the whole scheme of things, you actually have the subatomic world, which we will not be worrying so much about. So the area of my research is primarily focused on atoms and molecules. That's what we're going to be talking about today. So here's another picture of Ebola, and basically what you see here, and you don't have to read the word, but there's all these different proteins that are in a particular virus or in a particular bacteria or in our cells if we're trying to treat something where our cells are going wrong, and if we have a disease state, we need to understand what are the specific proteins that are in the body, or what are the specific proteins in the pathogen, and how do you actually interact with them. And this is the role of not what we're doing, but this is the role of biologists and a whole bunch of other scientists, geneticists who are trying to figure out what are the relevant drug targets that you can go after to cure a disease, for example. So Ebola obviously is in the news these days. The most recent chemical and engineering news has a very interesting article on Ebola that if you want to read about has a lot of good information. And they're starting to break things down and try to understand what are these specific targets, what are the specific proteins that are involved in the function of this organism. And if you could understand those particular proteins that are involved, then you could start to figure out how to disrupt their function. And then the other people that come into the game then after that are the crystallographers, for example. So once you, we heard a recent talk on crystallography, so once you get a sense of what the target is through biology and genetics, for example, you figure out the target of interest, and you can crystallize that target. And once you get a crystal structure, that essentially gives us a picture, a three-dimensional picture of what the target looks like. And that's where my group and our field of computer-aided drug design comes into play. Okay, so why are we going to simulate these atoms and molecules? It's nice that you have a picture of a protein, for example, and we could run simulations, and people have heard about things like folding at home, where you try to fold proteins into structures. So we want to understand, why do we simulate atoms and molecules? So does anybody know who said this? So everything that's living can be understood in terms of the jiggling and wiggling of atoms. Anybody heard that before? No. Thank you. Very good. So that's our good friend Richard Feynman, one of the great physicists of all times. And this is why we're running these molecular simulations. We're trying to understand how atoms and how their movements can be used to understand biology. It can be used to understand how the body functions, and how everything else functions. So there's various levels of complexity with which we can model these, and there are people. Yeah, the question back here.
What function do the proteins serve in a virus?
So what function do the proteins serve in the virus? It's an excellent question. So. Okay, so there's two different. Those are two different questions. So, what function do the proteins serve in the virus, and what makes the virus so deadly? So basically, all living things, and you can debate whether a virus is a living thing, but all of these, viruses and other living things, they're basically just trying to survive. Right? They're trying to survive and propagate, and they're not necessarily trying to kill somebody. They're not thinking about they don't like this person and they want to make them sick. They're just trying to propagate and evolve. And it happens that some of these things are very healthy for us, right? So we have all these bacteria in our gut that are very good. We've evolved to have a symbiotic relationship with these things. And that just happens to be how things work. We've evolved with them. And there are these other things that we haven't evolved with, and those things actually can be detrimental to us because they don't interact in a favorable way. Our body responds negatively to them, so we can get inflammation or fever, or they can break down certain cells or various other things in our system. So Ebola itself has various different proteins that do different things. It's a very, very, very simple virus. And you saw the picture there. There's something like 8 different proteins in there. Some of them serve to form the cell structure, the kind of the matrix that holds the thing together. Some of them serve to help replicate the host's DNA, and so that they can propagate and take advantage of that machinery. Some of them serve to just help with their cell cycle and the use of energy and getting into the cell, for example. So each virus or each bacteria or each cell has a bunch of proteins that serve these various different functions. Yeah, right there?
Is protein a type? Protein is a type of molecule, right?
Protein is a type of molecule, that's right.
Does it have a complex or a simple type of molecular structure?
So proteins generally have somewhat complex molecular structures. So proteins have three-dimensional structures that I'll show a little bit here, and they have what we call alpha helices that are these kind of spiral type things, and these beta sheets, these two things that lie against each other. And the combination of these alpha helices and beta sheets allow them to form three-dimensional structure, and it's their three-dimensional structure that gives them their function. And so depending on their three-dimensional structure, That allows them to move in different ways, it allows them to go to different parts of the cell, it allows them to interact with different proteins, it allows them to bind different molecules, it allows some of them end up being enzymes because of their three-dimensional structure. So their three-dimensional structure is relatively complex, and in a sense, evolution is a consequence of all of these amino acids coming together in these very complex three-dimensional structures. And as a result of them coming together this way, they serve a particular function. And in a sense, everything has evolved to serve a particular function, otherwise it wouldn't replicate. Okay, great. So let's talk about the various different levels of complexity. So we can see that there's, at the end of the day, we would like to have a very simple system with an on-off switch. And that's the way that we used to think about a lot of the work that we're doing. We have a particular protein, and we'd like to either turn it on or turn it off. In many cases, systems are much more complex, and you could still have a light that will tell you when you're on and off, but there's not a switch anymore. There's just a light for on and off, but there's all this other stuff that's going on here. So there's various levels of complexity all over the place. In our family, I'm up here, my wife is down here. We're all kind of familiar with that, right? So in life, there's chemistry and biology, and we're operating kind of at the intersection of chemistry and biology. Chemistry is relatively simple. We understand how atoms work. We understand the basic quantum mechanics that govern how atoms interact with each other, how atoms form structures. And in a sense, all we need at this point is just big computers to be able to simulate complex chemistry. And we still have to make approximations, and we still have to formulate our research topics properly. But we have a pretty good view of how to proceed with respect to chemistry. Biology ends up being much more complicated. Now, of course, at the end of the day, all of these things are related. You have atoms and you have subatomic particles that make up the atoms, and then the atoms come together and make molecules, and molecules start to join together to create cells, organs, and organisms, and these things are all related. So there's no real line that defines the intersection or the end of where chemistry ends and biology begins, or when biology ends and genetics begins, and when genetics ends and physiology begins, for example. And so we really all have to understand this whole cycle. They're all part of a continuum, and that's what we're starting to see happen in the universities more and more, and research more and more, and we have more of this cross-disciplinary studies that are going on. So in my research, we're mostly kind of on this side. We're dealing with mostly atoms and how atoms come together and make molecules, and how molecules then come together and interact with each other, or how you disrupt their interactions. And then there's this whole space, and once you understand how those things come together, you can start to affect cells and cell function and organs and organisms, etc. So like I mentioned before, my background is physical chemistry, and that's what we're doing on computers now. So we're studying how all these atoms and molecules interact, what their energies are, etc., using basic rules of physics to simulate them and computers. So we focus on the intersection basically between chemistry and biology. We rely on experimental biology to determine targets, and so we can't simulate a cell, for example, to figure out what's important. We have to work with a lot of collaborators to figure out what's important. We leverage a lot of physical chemistry to model the interactions, and we use a lot of computers to try to understand what's going on here. So I want to just talk a little bit about what it means to use a lot of computers here. So at Schrodinger, we have thousands of computers. We have about 5,000 computers in our cluster, which is a good number, but it's actually still not that much. We've started to run calculations now on graphics processing units, so people may be familiar with these things for playing video games. They're kind of specialized. Everybody's got a GPU in their laptop. Everybody's got a GPU in their desktop. There's high-powered GPUs that some of you older kids might have for playing video games, because if you don't have a high-powered GPU, the graphics kind of get slow and sluggish. We're in the same situation. We don't want our calculations to be slow and sluggish. And so we get these very high-powered GPUs, and it ends up that these types of processors are very amenable to certain type of scientific calculations. And we have a class of calculations that we run, molecular dynamics simulations, where we can leverage these GPUs, and one of these little GPUs provides the equivalent of about 100 normal processors. So we have machines that have about 8 GPUs in them, which is a very simple machine that you can put under your desk with 8 GPUs in them, and it's equivalent to a 1,000 processor cluster. So we're really getting this huge benefit in a lot of the modern technology that's being developed completely independent of science. These GPUs NVIDIA and these guys developing these things are not focusing on the sciences. It's a benefit to them. They want people to play video games. They want people to play very, very good, high-quality 3D video games that get them sucked in and want them playing more video games. And we can leverage this to actually run these very high-powered computer simulations. Now, when these are not enough, and this is what we have in-house, this is what we have in our offices, we can start to run calculations on the cloud. And the cloud people have heard of, so Amazon has a cloud, Google has a cloud. This is all those computers that are sitting out there in storage, big storage facilities that basically just have a bunch of computers. Schrodinger actually has 2 of those in Schrodinger, the company I work for, if I didn't mention that previously, and we run a lot of these simulations. We've actually run the 2 largest Amazon cloud jobs ever. We ran 50,000 computers simultaneously running all over the world. We have computers running on 5 continents simultaneously. 50,000 CPUs running simultaneously for a drug discovery virtual screen. And then more recently, we run 150,000 computers running simultaneously, or more for a material science project where we were looking at how to screen for better blue OLEDs. From our perspective, we've been working on, and I'm talking here primarily about life sciences, about curing diseases with code, but from our perspective, we don't actually really care too much about whether we're looking at material science problems or life science problems. Either way, it still has to do with atoms and molecules jiggling around. When you move beyond the cloud, excuse me, kids. Are you guys listening here? Are you? Okay, thank you. This is my daughter, so I can call her out. All the rest of you have to take care of your kids. If you need more computers than 100,000 computers, for example, which is what the cloud can offer, there are other options. For example, Vijay Pandey, who's at Stanford, he does folding at home, has a very interesting setup where he has folding at home, and any of you can sign up for this, and you can actually run molecular dynamic simulations on your home computer. Many of you may have already been signed up for this. And this will allow you to run these types of simulations, and they run millions and millions of independent, very short simulations, and they have a very intelligent algorithm for how to pull all that information together into one coherent simulation that obeys all the laws of thermodynamics, but has been done in a very piecemeal way. And they have access to millions of personal computers. And then there's also very specialized hardware, for example. So, for example, the D.E. Shaw Research Group, DESRES, who was actually an early investor in Schrodinger, has put in a huge amount of time and money and resources into developing a very, very, very specialized chip that's designed just for running molecular dynamic simulations. These computers here that you see can't run Microsoft Office. You can't surf the internet with them. You can't play video games even with them. They're just for running these molecular dynamic simulations. And each little bit in this whole processor has been designed very specifically just to optimize the process of trying to get as many steps as possible for running a simulation. And don't worry, I'll show you guys what a simulation looks like in just a second. And so these guys here can start to run microseconds of simulations per day, or tens of microseconds per simulations a day. That's hard to understand exactly what a microsecond is. I see some people in the audience saying, wow, that's a lot of time. And I see some people saying, microsecond, that's like faster than I blink my eye. And both of those are right. A microsecond is a very, very, very short period of time relative to what we're used to. And at the same time, for a simulation, you can start to see interesting events happen. Microsecond is kind of the time scale where you start to see proteins folding, for example. Fast folding proteins can fold in low